Kira, welcome to the Southern eDNA Society March webinar. We're very pleased that you could attend. Today we've got two very wonderful speakers, Dr. Anastasija Zaiko and Dr. Maarten de Brauwer, who will be talking about some of their really cool eDNA work. As we're in New Zealand, we'll just start with a quick karakia. Whakataka te ho ki te uru, whakataka te ho ki te tonga, kia mā kina kina ki uta, kia mā tara tara ki tai, e hi ake ana te atakura, he tio, he huka, he ho ho, ti he Māori ora. That's just a little traditional New Zealand opening to wish us a successful meeting. We've got a couple SeDNAs announcements. So the newsletter went out about a week ago. Maarten did a great job on that. So be sure to check your email inbox if you've missed it. We're working on progressing the Knowledge Hub, and soon we'll be looking for volunteers. So if you've been looking for a chance to get involved with the society, this is a really great opportunity. I'm sure if you email the hello@sednas. .co.something.
It's on the website.
It's on the website. There you go. Yeah. Feel free to get involved. Secondly, there's a photo contest. So win a free PeerJ publication, which is quite a lot of money. So consider doing that. We've been seeing some really cool pictures come up on LinkedIn, Blue Sky, Instagram. Tag SeDNAs using the hashtag. And yeah, there's an updated deadline for that as well. That was an old deadline. If you're interested.
Is it truly till the end of January, Claire?
Updated and is end of April or end of March?
Yeah. I think it's either the end of April or March. Either way, get your photo in as soon as possible. And then the conference organization, led by Erin Hahn and Diane Gleason. If you're interested in helping out with the conference, get in touch with Erin at her email there. Just a few little webinar announcements. Webinar sponsorship is open. So if you want to get a little bit more recognition for your brand, feel free to let us know and you could get your logo up on these webinars. Our next webinar, I don't have anyone specifically rostered. So if you do want to talk, please do get in touch. It's a really nice and friendly way to go and chat to people about your work. Yeah, you'll get some really good engagement. Just highlighting a partner webinar that's coming up. So that's Wednesday, the 1st of April with Australia Biocommons. And there'll be more information on the website soon. And just highlighting that past webinar recordings are up on YouTube, and then future webinar recordings will either be on YouTube or the Knowledge Hub. One thing that we do want to highlight, we want to give some shout outs to our members. So if you know a cool member doing some awesome work, let us know. We'll give them a shout out here on our webinar. We're highlighting Dr. Amy Vanderies and Dr. Richard O'Rourke, who are doing DNA Drive. So this helps look at bugs and insects and other DNA that you can get on license plates. So they basically asked people to go out, drive around with a license plate, and then get some eDNA off of it. So we're super excited to see those results. It's been highlighted by RNZ over here in New Zealand. And yeah, kudos to them. And I'm sure we don't need any introduction, but we're happy to hear Dr. Anna Zaiko and Dr. Maarten de Brauwer talk about their research. All right, let me stop this share.
Okay, so do I start first, Claire?
Yeah, if you could share your screen. Let's see if
now. Okay, please let me know if you see the screen, the presentation mode.
Yep.
Yeah, looks good.
Okay, perfect. So I start with a little pepeha, telling you about myself. It's like a New Zealand Aotearoa tradition. Kia ora, ko dana te awa, ko te moana patchy te moana. no rituania ahau, he whakatu ahau e noho ana, ko Anastasija Zaiko toko inoa. No reira, tana koto katoa. And today, and thanks again, Claire, for organizing this, restarting this webinar series. I think it's a great initiative, and thanks for SeDNAs for hosting these webinars. I'm really glad to present today and talk to you about some potentially new ways for eDNA community, how we can collect eDNA from water. And what I want to mention that although I'm presenting here on behalf of Sequench team, there's like a contribution of the entire team to this work, and big kudos to them because they all helped in some way or another in this development that I'm going to present today. Okay, so as I said in my PPH, my name is Anastasija Zaiko, you can call me Anna as well, and I'm originally from Lithuania, now living in New Zealand. And on this beautiful picture with a shark, I'm actually sitting on top of the shark together. It's like the photo is taken together with my colleagues from Lithuania, Benthic Ecology Group, and it was in the Red Sea when we're doing some like fun dive stuff over there. And by training, I'm an aquatic ecologist, so I just started like working in the molecular eDNA field only about like 15 years ago, but I was like working in the ecological research all my career. I'm doing a lot of biosecurity, benthic ecology, aquatic ecology in general, and currently I'm a co-founder of Sequench company and also president of the Southern eDNA Society. And this is my hometown, Klaipėda, and actually the view of, at the university campus looks beautiful. You can see a little bit of light blue water in the background. It's Kronian Lagoon and the Baltic Sea. And for those who don't know where Lithuania is, it's like a tiny country in the center of Europe. It's even smaller than New Zealand. The population is less than 3 million, so it's really a tiny country, but beautiful, and like really invite you to visit it one day. And no surprise that living like close to the sea, I was always attracted to water. That's how I became aquatic ecologist. And 12 years ago, me and my family, we did a big move from Lithuania to literally another side of the globe, to New Zealand, and where I started to work at Cochrane Institute, the biosecurity group there, and shifted like from doing mostly like ecological, biological research to more working in eDNA projects, and met a lot of very like greatest scientists in that field, and that learned a lot during this journey at Cochrane here. And 5 years ago, I again did a big shift from academia to like private consultancy business, and together with my partner, we opened Sequench company that originally was planned as like service provider, sequencing company, working like purely as service provider for other organizations. But then we realized that there's like kind of a niche and demand on like a wider services and wider collaborations. So we started to work with other organizations, like providing some sampling solutions, helping with study designs, library preparations, data analysis, and environmental consultants in general. And our specialty, I could say, is more like bespoke services to people who are interested in molecular research and eDNA, and we are happy also to collaborate on research and development projects. And my whole idea was that I wanted to keep this scientific component in my professional life, so we do still work a lot in the research projects. But when we started like to work in that space more and more, we heard like many questions from our partners and clients, like, you know, I have this challenging project, how do I collect samples? And in some cases, we had like really good solutions to offer from those available on the market. And in some cases, it was really challenging because not like I would probably struggle to say like if there were like 2 or more than 2 projects that were absolutely identical and the same, because all of them, they're so different, they're so different requirements. Some of them truly challenging, like in terms of samples to be collected in challenging remote environments. So we started to think, okay, how can we help people to approach this sampling challenge in a more efficient way? And for those of you who work in eDNA field, you know that there are many ways how we collect samples, and because eDNA is so versatile method that you can sample from water, sediments, feces, air, anything literally. And you can use like simple, like traditional collection tools, basically collect samples the same way as other biologists, ecologists do in water from sediments using this like bulky, logistically heavy equipment that works pretty well as well. And also, more recently, there are more and more tailored eDNA sampling approaches appeared, and these range from very simple ones when you just like need a syringe of water taken from a river or a pond, and you push this water through a cartridge, a filter cartridge, and you concentrate your eDNA sample this way, to a more complex ones where you have like a in situ pump systems or like even automated devices that can be deployed to like big depths in the ocean or left unattended for weeks or months, and they can collect samples for you. And also some creative ways of collecting samples, like just to realize that sometimes we just need to deploy some substrate into water and eDNA will accumulate passively on it. So all these different approaches, they work in different like ways, and they also work pretty well for different applications. But of course, they are like in some cases, they are not enough. And especially what we noticed that although we keep saying that eDNA sampling for water is effective and easy, and this is like the main message that we try to convey to people when we want to convert them to eDNA methods. And this is true, in fact, in many cases, that's like how it can be done. But what we are facing more and more often that in some cases, in many cases, in fact, you need actually to be able to effectively concentrate quite large volumes of material or like concentrate eDNA over large areas. And here I'm talking predominantly about aquatic ecosystems because that's my kind of like the area where I work the most. And this might be quite challenging, in fact. And the overall concept and message that we hear like more and more often, that larger volume and area coverage is really important to enhance biodiversity capture in general, and also specifically for detecting rare targets like invasive species in the initial stage of invasion, or like rare protected taxa. And imagine that we need to sample really large systems, like an open water system, an ocean, or a big estuary that Maarten will talk about a little bit later, or like a big lake. We want to upscale our sampling program. So it could be quite a big challenge, in fact, to find an efficient solution. And especially if we start considering such limitations as time constraints, because if you're running a big, like large scale program, sometimes you do not have enough like station time, or you want to feed multiple locations in one day, and there's like it could be a big challenge to address it effectively. Budgets, we all live in this context of shrinking budgets and resources, and we always want to find solutions that are cheaper, that are cost efficient and affordable. If you want to involve people who are not professional scientists, and we use this more like opportunistic approach, again, like addressing this time and budget constraints, sometimes we go to like, and it's getting more and more popular, involving citizen scientists in the research. We want to have something that is reasonably lightweighted, that is also quite easy, and that doesn't require substantial training, and that can be handled by non-professionals, in fact. So after a lot of thinking, and also like listening to this, like getting these questions from the colleagues and clients, we came up with this kind of wish list, how we would imagine this, what features this sampling system from the open water ecosystem should look like. And you see there is quite a bit of requirements that we have listed. And many of the devices that are readily available, they can tick some or many of those points, but we've thought that actually none of them quite ticks all of them. And then we decided, should we try and actually challenge ourselves and develop something that would work for these like more complex cases specifically in the open water ecosystems. And we came up with this device that many of you have seen probably at the eDNA conference. It was presented there, like it actually was the first like working prototype that we started to deploy and test and in multiple like pilot studies. And here in this short video, I'm presenting the new like second version of it that has been improved, addressing a few feedback responses that we received from our first adopters. And the whole idea is pretty simplistic. So it's a towable, deployable device that can be towed behind a boat, any boat, in fact. At high speeds, it can operate at speeds from like literally about 4 knots to 14 knots. And the concept is quite simple. So it's like a hollow tube with wings, and it utilizes simple physics, a Venturi effect. Because of the narrowing in the middle of this tube, it induces suction from the side openings where we insert the filter cartridges. So it just uses it. There is no pumping system. There is no like any complex hardware. It's like really a very solid and robust piece of equipment. And it operates with these simplistic filter cartridges that we also developed for this device, but then it can be actually adapted and adjusted for other devices as well. And the collected sample can be preserved by a default desiccation function that comes with the package of the filters, or it can be also preserved with the buffer solution or frozen. And the whole idea is that, it's a simplistic approach for collecting samples where scale and operational simplicity matters. And also alongside with this system, we developed, as I mentioned, these filter kits that are also quite simplistic solutions. So it's a filter membrane holder that is made of compostable plastic. And it holds a standard 47 millimeter diameter membrane. We started with 20 micron membrane, but then now we offer also 5 micron option. And it is preserved either by desiccation, as I said, or it can be also used with the buffer or frozen as well. So in fact, this system kind of ticked all of or most of the points here. But then we also looked in this last point that was a little bit more challenging to address. We wanted to make it really streamlined, integrated into workflows. And although it's not like it's still a operator handled device, but we wanted to simplify at least some of the steps and allow some kind of automation. At least if we cannot automate the sample collection, we thought, could we automate the metadata collection with this device? And here, actually, for the first time, I present this feature that we are developing now. And we hope that it will be ready to offer with the device in this filter kit a little bit later this year. So it's the smart filter concept. And the whole idea is that we are developing an app that will automatically record all the information on the metadata of the sample collection, like the tow parameters that will include the coordinates, time, also will have a timer for the tow. It will calculate the estimated volume that goes through the filter openings based on the speed of the boat and also the towing time. And the beauty of it that we will introduce a small recording chip into the filters. Then after the recovery of the filters, the information on this metadata information will be transferred directly to the filter and recorded on the filter itself. And then when in the lab, you can just like scan the filter with the simple NCF scanner, or like that's available on many of the mobile phone applications, or you can have a dedicated scanner for that. And then it can be easily integrated into the CSV file with the sample metadata spreadsheet. And then after that, you can use it for label printing or just like tracking the sample processing steps further down the track. So it actually simplifies one of the challenging, like it's not probably the most challenging steps, but not the most exciting step of the fieldwork, like the metadata recording, and also you doubling up and have this redundancy of the records that is held on your mobile app device, but also is recorded physically on the filter. So this is kind of an exciting addition that we are developing right now. And also another couple of things that I wanted to mention, that although our initial development was related to this yellow submarine, the filter itself, then we realized that actually we can use this filter in many different other applications. And we designed this like stick pump device that can be used as a point sampler, like, let's say in freshwater ecosystems. And the whole idea is to maximize again the volume that we can collect and concentrate on the filter, and also allow like very fast replicate sample collection with a pretty simplistic device. And also it's quite an affordable solution. So you can collect 4 samples in one go with the flow rate of 45 up to 45 liters per minute. So basically you can have in 5-minute time, you can have up to 4 samples with 100 liters concentrated on them. This is, we have tested it quite extensively in different like settings in different ecosystems, and it showed that it really helps like to detect rare eDNA in freshwater systems, but also simplifies the work of the field teams a lot. And just another like a small little glimpse on another system that we have developed in cooperation with the citizens of the SEED program. And this one is specific for another very niche application, the collection of samples on racing yachts, like kind of a citizen science vessel of opportunity project that allows a very simplistic collection with the minimum manipulations on board, and also very robust system that can be fixed easily when in transit or being far away from the shore. And just like a cool picture showcasing where these devices have already been tested, and kudos to our first adopters and testers and prototype testers who contributed a lot with the like validation of the devices, comparing them with different other methods, and also providing really valuable feedback that we try to implement and like improving them and keep improving them for the future. And probably the final message that I wanted to emphasize, that one size doesn't fit all, and eDNA, because eDNA is a method that is so versatile and there are so many applications of that, you need to think carefully and think like of what is the best way to collect your samples that suits your project and your problems. And of course, our devices will not solve every sampling scenario, and there will be multiple different ways how to collect samples. But our idea is to focus on the current gap that we see that other tools cannot cover currently, make large volume, large scale eDNA collection robust, practical, and accessible. And to finish, I want to show like a few nice pictures with people sampling with our devices. And sometimes I feel jealous because our yellow submarine traveled to exciting locations where I have never been myself. Thank you, guys.
Amazing. Thank you so much for sharing about all of your cool inventions. If anyone has any questions, please feel free to pop them in the chat. I'm sure we've got time for 1 or 2 before we kick up. I can see Faith has raised her hand. Let me see if I can yep. Faith, would you like to unmute and ask a question?
Yeah, good. Can you hear me?
Yeah, we can hear you.
Yes, please. Good morning. It's morning over here in
Oh,
Ghana.
wow.
I'm a Nigerian, and I've actually joined SEDA Society for I think about 2 years now, though I'm inactive. I see eDNA as one of the aspects that you can use to understand the environment generally. So I've been inquiring about eDNA since 2021. That is like 5 years ago. But unfortunately for me, I'm not able to get hands-on experience until last year. I was able to work on eDNA, using it to get more data on harmful algal species and coastal marine environments in Ghana. But my first question is, in your group, your research group, are there opportunity for students for internship? And then number two, the patent equipment, are they available for like let's say a survey, a biodiversity survey? Like if I want to carry out a survey in Ghana, is there a way I could get them just to use them for now, that while it's still patent? I don't know if something like that can be done, like a trial in another environment, not necessarily where where it's been invested. So aside my question, the presentation was very wonderful. Thank you.
Thank you, Faith. I don't know, maybe Clara, you could address the first question about the
Yeah, I don't know about internships and things like that. I know that the SeDNAs website does have opportunities listed. I believe that there is a master's scholarship doing some eDNA stuff, and a few other opportunities as well posted on the website. But that would probably be your first point of call. Alternatively, I know that there's a lot of really nice and keen people here. So I'm sure if you found somebody that you wanted to work with and shot them a message, an email or something, they might respond. Yep.
And yeah, and for the second question about the availability of the devices, so yes, they are older. They are developed here in New Zealand, and they kind of went through this one or a little bit more than 1 year of these testing validation studies, but they are being used already and being purchased and used on different locations for like monitoring and like research purposes. So if you're interested, yes, they can be available in Ghana as well. It's like a little bit of this like figuring out how to send them there, and they're really easy. That's pretty small. The yellow submarines like under 2 kilograms, and we send it usually by DHL to different countries, and no problem at all. And they're also, like as I said, they're quite affordable. Both devices, they're under like the yellow submarine, the pump, the stick pump, they're under 2,000 New Zealand dollars current price. So it's like about 1,000, below 1,000 US dollars per piece.
Okay, thank you so much.
Yeah.
I see there is a couple of questions. Do I have time to answer those like 2 quick questions in the chat?
Yeah, I think
I can write it in the chat as well.
Yeah, there's 2 questions in the chat and then one on the Q&A. If you're able to answer those, in the interest of time, we might move on to Maarten.
Okay.
But yeah, and then we can have a larger discussion if there's time at the end of the webinar.
Is this one sharing successfully?
Yeah.
All right. Hi, everyone. That was a cool presentation, and it's always nice to see the exciting stuff that's happening. We've actually tried we've dubbed it the whippersnipper, and it works remarkably well. There's a lot of water going through. It's pretty cool. It's just unfortunate that we tried it at the end of our big project, so it just felt a lot more painful to filter water with a syringe afterwards. Anyway, Before I start, I also would like to acknowledge the traditional owners of the land on which the research that I'll be talking about has been done. And I'd like to pay my respect to the elders past and present. So what I will be talking about is a quite large-scale monitoring program in New South Wales, Australia, in the estuaries of that state. So let's kick in. Just a bit of background on the project. So the reason it got started and I was asked to design it was after the big floods in 2022-23, the government basically realized we don't have any real baseline of what lives the biodiversity in the estuary. And so under what I'll be referring to as NEAP, which is the New South Wales Estuarine Asset Protection Project, we started an estuarine monitoring program. And really the goal of it is to create science and to create biodiversity understanding that's relevant for management. Important, it's a very collaborative project that we're talking about. It's not just myself. There's multiple universities involved, multiple government agencies, and importantly, a lot of traditional owners have been working with us as well. So the main objectives of the monitoring project, the core thing is we wanted to re-establish a baseline of what lives in the estuaries in New South Wales. So it's not just eDNA that we're doing, though eDNA is definitely the core method. And then a big part of it was figuring out which methods are cost effective to monitor estuaries, which should have their own challenges compared to, say, marine or freshwater environments. And to finally, for the government, develop a framework of what a future long-term monitoring program could look like. A big part, it started small, but it's been growing and it's been a joy, is Indigenous engagement. So at present, we've been working with 13 groups across the state. You see some of them over here. And it's basically beneficial for both the groups that we work with, but for me as well, and for the team, it's pretty useful because the local rangers, they'll get employment opportunities, they get training in scientific techniques, and they feel empowered to get a better understanding of their environment. For us, it's great because we get local capacity and understanding of the areas that we work at. It's less travel costs. We need to send far fewer staff out. And it gives us, and especially for government, more important, I guess, than universities, a bit more social license to do this research on country. This wasn't an easy project, and I really want to acknowledge the cultural restoration team underneath, which is linked to the Marine Estate Management Strategy. So that's Ian and Kelly. And basically, all of our connections have gone through their teams. It's kind of like a funnel where they work as a centralized, trusted contact to the different groups across the state, which was really crucial because it meant, importantly, that the groups are interested, so we were not overloading random groups with cold calls. It meant that Ian and Kelly, they understand the capacity of different groups, whether or not they'd have time and experience to work with us, and it ensures a lot of fairness as well. So we, especially myself, as someone who at that point, 2 years ago, wasn't very familiar with the landscape here, it's kind of easy to go to the more visible groups, but working through people that have the experience was really useful because we could really reach and make sure we got a good spread of groups that may not be as much in the public eye. I do want to acknowledge Kate, who's on quite a few pictures here, who's been my main colleague, and then she's been very much responsible to making sure this works like a dream, really. So it's been really, really good working on this aspect. All right, to the project design. So when designing this project, we'll get to eDNA soon. There's a lot of things to balance in how you look at it. So there's things like which places have been flooded, are there existing government projects going on, the geomorphology, which is basically, is it a large bay, is it a small creek, disturbance level of estuaries. There's a big latitudinal gradient from the subtropics to temperate zones. And then there's a lot of logistic questions as well, because we've been using video cameras and sediment sampling and quite a few other methods. So balancing that all off in the design was pretty crucial. And it comes with challenges. So you see all the sites here in New South Wales. There's a lot of logistics involved. There's a lot of it's pretty pricey, so we luckily had a good budget for this. And key here, and this is relevant for the eDNA, is method suitability, because we work with non-experts quite often. It needs to be easy to use, it needs to be safe, and cost efficiency is pretty key. Another challenge we had was weather. Halfway through the project, we had a cyclone hitting the north coast of New South Wales, which led to floods. So we then had to go in and do impact assessments there as well. So that was kind of a nice little treat on top of it. To zoom in on the logistical challenges, just to give you a bit of an idea and a feel of what it was like. So in the last 2 years, less than 2 years, we traveled over 35,000 kilometers up and down the coast. We spent with the whole team more than 150 days in the field. And Kate and I did a count yesterday. We've taken 108 people out in the field doing eDNA sampling. But the core team are the three Muppets you see below. That's myself, Kate, and Isa. So that's the core team that's been leading the program really. And one of the other joys, because it was both fisheries and university, is that we got to do double the paperwork for everything, the risk assessments and the ethics and stuff. So it's been quite fun. So what it looked like, the final design is 9 estuaries, the ones in yellow here. They were done in depth, sentinels or in-depth surveys, and they were done twice a year. So we've got 4 replications there, and they've got 9 sites per estuary. And what we do there, we take eDNA samples, we take sediment samples, and we take samples so the sediment is for macroinvertebrates and for pesticides and other pollutants. On top of that, the purple dots, they are surveillance estuaries or rapid ones, which are done once a year. So we've done once in winter, once in the start of the project, once in summer at the end of the project. But we only do eDNA in those sites. So there's no sediment playing over there. And then because of Cyclone Alfred, we did the northern region, which is 11 estuaries, twice on top of that. So just after the floods and 2 months after the floods to see whether there was any recovery from the impacts. What we did in terms of the eDNA, I'm not going to go into the other methods too much. Because of all the restrictions and the large scale, we went for very easy citizen science-based kits, basically, of the Wilder Lab and did Tree of Life metabarcoding on this. To give you an effect, the last baseline for estuaries was taken in the early 2000s and was just fish. So getting the whole picture 20 years later or 25 years later is pretty useful here. So let's have a look at the results. We only just got back from the field less than a week ago, so there's not too many in-depth results yet. So there's a brief overview, let's put it that way. So the eDNA summary, we've taken 3,700 plus samples in the last 2 years in 41 estuaries, a lot of sites, 102 sites in 6 different seasons, so 6 temporal replications, which means that we've basically got 589 sampling events, as in sites that are sampled over time. So quite an extensive amount in 2 years. We've detected over 10,000 taxa to this date, and you see there's some differences in the different regions, which you see on the map in different colors. So there's a bit of a trend there already that we'll definitely be diving into, because this is not taking into account of the seasons yet. Just for the backup to give you, again, what else we have in that data set, we've taken, each time we went out, we take water quality, so there's things like pH, dissolved oxygen, temperature, at 81 sites, so those core yellow sites, the sentinel sites. We've also taken macroinvertebrate sediment cores, which were mostly processed by Isa on the picture there. So it's looking at like little shrimp and mollusks and so on. We've taken pesticide data at 81 sites and grain size characteristics as well. So all combined is going to be really, really exciting to look at how that all relates back to the eDNA data. I'll start more traditionally, some of the things we've detected in the eDNA. The classic thing, like we've obviously done this, we look at threatened and protected species. I think we are at about 30 protected species now. So it ranges from little birds. This is something called the Verixitella, which was only detected in a national park in the Sandon River. Quite a few seagulls, white-bellied seagulls, which are also quite an important totemic species for some of the groups that we work with. We got great white sharks in Nelson Bay, which is always fun to find those. And we've got platypus in a few areas. and quite a few fur seals. I unfortunately didn't have time to add the video. One of the lower sites there in the Biga River, we detected fur seal, which is threatened, but we've also detected invasive species, the carp, which is a big issue there. But while we were sampling, we actually saw fur seal eating a carp. So it's pretty cool that you get to see things that threatened species are eating pest species, which is kind of nice. So that was fun. Then biosecurity, we've done like the good and the bad. So we detected over 70 different pest species by now. But the cane toad is quite exemplary because they are starting to march south into the state. And so what we found in our surveys early on is that we obviously detected cane toads in the area where we knew they were present. So that's in the northern part of the state. We unfortunately detected them in the buffer zone, which is, for people familiar with the state, between Yamba and Descendant, where they shouldn't be present, but we detected them there. And we have since detected them in the cane toad free area, the super free area, so they're there as well. So this data, well, I haven't published any of this yet. We connected straight with the government agencies responsible, so that's biosecurity, DPID, and local land services. And they've been following up, and the last news I had from LLS, local land services, they are now doing a large sweep in the buffer and cane toad free area with eDNA, with qPCR methods, where they're looking at different dams on properties to see if they've got more cane toads there or not. So there's this straightaway management action. So you don't, as a scientist, like, this is what I find exciting. We don't even have to publish to already have a management effect, and that's really exciting about this project. We can do more traditional ecology. So these are the more in-depth estuaries, and so you can look at things like species richness and where they lie in the estuaries. So at the bottom, you'll see the 9 estuaries from north to south. So Richmond River is the furthest north, Biga is south, and the richness on the y-axis. And so what you see is traditionally, and that's what you'd expect in ecology, is that closer to the mouth, to the ocean, you have higher species richness, whereas lower, closer to the freshwater, it gets lower, so you get less species. And the interesting thing is Biga River stood out as the inverse. And this baffled us until we went to chat with the ranger group there, the local Aboriginal Land Council, where we had a good chat. We talked about results, and I went, honestly, I don't know why, for some reason, your freshwater sites are so much more biodiverse. And one of the more experienced rangers just went, oh, it's obvious, because that place where you collect it, that's the drainage of a massive wetland area and a swamp. And so when you look into it, it's most of the species there are wetland species that otherwise we wouldn't get. So it's something I would have struggled to find. Maybe I would like to think that I would have discovered this in the end, but just a simple conversation with the local guys was straight away that was solved. And this touches upon something quite important when we just, with Tree of Life method barcoding, only use species richness. And I love this graph to illustrate it. So what this shows is different types of estuaries. You don't have to be an estuary specialist. What it basically means, there's different categories of estuaries, and some are very large, like bays or drowned valleys. They're kind of large. Creeks are smaller. And if you purely look at taxonomic richness, there's a bit of difference, but it's not very satisfying for me. Like, I looked at it, I'm like, oh, that's not really much. There's something going on, but not super clear. And that's just richness of Tree of Life. That's all the taxa, right? But then we started looking like, what are we really interested in? What is management interested in estuaries? And partially triggered by that conversation with the Biga mob, they basically, like, we designed something what we're at the moment calling a marine affinity score. So it looks at the identity of the taxa, and to go whether or not they're marine or they're mixed marine, they're brackish, or they're fully terrestrial, because we do get plants and so on. So in this case, a plant like a tree would get a 0 because it's fully terrestrial, whereas say a tuna would get a 1 because it's fully marine. And when we plot that, the score per geomorphology, you get a very, very different picture. So then you straight away see they're different, and this ecologically makes a lot more sense to me, where you can see things like a bay is much more marine driven versus a creek, which got a bit of everything, but it's quite lower. And then suddenly it starts making ecological sense. And if I can, like, one of the big takeaway lessons I've had from this is you can't simply go, richness is everything when you do tree of life metabarcoding, because it dilutes the actual ecology of it because it's so all-encompassing. You can do the opposite. So I've looked at trends with terrestrial affinity, so more or less terrestrial, and you can see trends that you'd expect, right? So the further away you go from the river mouth being at the marine, and then the further up you go the further upriver you go, you get more terrestrial influence. It might seem logical, but it does mean if you look at species richness that's higher further away from the mouth, you need to incorporate the fact that there'll be a lot more terrestrial species in there. And if you're not looking for terrestrial species, that's an important thing to be aware of. What we see is an opposite trend, again, pretty logical. The further away you go from the shore, the less effect from terrestrial species you get, pretty logical. But then after the floods, we were thinking about the freshwater influx. So what we see is if you get more rain, so this is average daily rain in the week preceding sampling, that definitely increases your terrestrial affinity score. So depending on what your question is, if say you want to look at terrestrial species, you actually want to go sampling in a river after lots of rain. If you're only interested in what lives in the river, you might actually want to hold off from sampling after rain. So it's quite a cool result, I find, and we're working on a first paper to come out that works around this and see how different factors influence our results. And then we had the floods. So the cyclone floods. I've got a student, Simone, she might actually be here, and she's got a whole heap of amazing plots. But they're too good. I'm not going to include them because they're a bit too complicated, and I'll let her talk to it. So I've just taken one river. This is the Clarence River is up north to show this PCO plot. So the further the dots are apart, the more different the assemblages are. So the lower ones is close to the river mouth, the asterisks, the stars, they're nearly freshwater. So the first sample was in the winter, nearly 2 years ago. Then we did sampling in summer, and what you see, there's a shift in assemblage a little bit, but you see the same kind of trends happening. But then just after we took these samples, the floods hit this system, and we went 2 weeks after the floods. It was actually still flooding at this stage. There was like big branches, and at one point a dead cow floating by. And that's what your samples look like. So suddenly everything gets condensed, everything sits at that fresh area, and it's just one big jumble, and you really don't see any pattern anymore. So we went back 2 months afterwards, and what you see is recovering. It's more condensed than the initial samples, but it's recovering. And then another few months after that, we went again, and you see it starting to normalize and change again. I haven't included the summer 2026 samples, but it'll be very exciting to see what happens afterwards. So that's, for me, a very, very exciting prospect. And hopefully in a couple of months, we'll actually submit a paper on all of this. Another big question, and this is linked to the floods, a lot of the results are driven not just by our fish, what obviously we work with the Department of Fisheries, what they are interested in, but also by an unseen majority. Up to 40% of the texts are bacteria or single-celled organisms. And there's things like mycetes, which I'd never heard of before, cryptomonads, ciliates. So they make up a whole lot of the richness, the diversity we've got in the estuaries, but we don't really know how to interpret, well, I don't really know how to interpret that data and what that means to management. So that's things we need to work at. By the way, mycetes they're linked to fungi that cause the potato blight in Ireland, which something new I've learned. So that's something definitely we should be working on in the future. So what actually do they do, and should we be looking at them? Looks like we should. Starting to round off, so because we are working for management, and the opportunities to use eDNA in these kind of big, large-scale projects, obviously the cost is less than when you're doing more labor-intensive projects. We do it really quick, like we've sampled in, I think this season, the summer season, one of the quickest yet. In 2 months, we've taken over 1,000 samples. We've done the whole state in 2 months, basically. So it's easy to use, it's quick. So that's, for management, really, really useful, obviously. And you have the traditional stuff. We can do pest and threatened species, but you can also do at a whole different range of other applications that I've got data with shark teams. We've got seagrass research happening in some of our data. But there's also weird things. One, the Shire of Wollongong, they got back to me because they did follow-up surveys. They were inspired. They did some more, and they noticed that a seafood restaurant in one of their areas, around that area, they detected a whole lot of oceanic fish, and the restaurant is definitely not oceanic. So basically, they assumed that potentially they'd been illegally dumping their rubbish. They sent compliance out, and in fact, they had been doing that. So while it wasn't designed for this at all, it's one of these uses I wasn't predicting. But the Shire the council was very happy that they discovered that. There's obviously a lot of challenges as well. One of it is familiarity with the methods. Some people are very much ahead of the curves, others not so much. Some people have only just heard of eDNA or haven't heard of it at all. So that definitely can be a challenge. And then standardization, how do you compare it between different areas? In case for my program, it's easy, it's across the state, I'm organizing it all myself, it's standardized, but how does it differ between different states, or when a different council wants to use a different method, how do we standardize that is obviously a challenge. The big question, and this is by far the biggest challenge, is what does it really mean? It's easy to go, well, we detected a pest species, that's easy, but these large quantities of data that we get, what does it mean for management? Like, are the eumycetes, is that a crucial thing we should be looking at? I'm not really sure. So that'll be the next step in eDNA for management, really. And it's really that interpretation. So how do the functions of the species that we detect, the trophic levels, how does it all relate to different management objectives of different councils and government departments? Where do we go from here? So all our fieldwork is done, so we're onto paper and report writing. The end of the program as it is in 3 months. So end of June is the end of this program, because we were disaster relief funding. So at the moment, we're basically looking at funding to keep the research ongoing and to see where we go from there. So that's a story yet to be told. What I would like to do in the future with this program or the result is really improve the practical use of the results that we have. So to conclude, we've got a lot of large-scale biodiversity data coming out. There's a whole lot of applications for it, and there's a lot of exciting research to be done. I'm always open for collaborations. If people have ideas or they want to work together, just feel free to reach out. And that is it for me.
Great. Thank you so much, Maarten.
Okay.
If you have some questions for Maarten or Anna, please feel free to pop them in the chat. Anna McDonald, I can see, has put a little note for Maarten in the Q&A. If you could just maybe talk a little bit more about the analyses.
Let's have a look. So Anna, good to see you back from Antarctica. This has not been published at all, actually. So we've been, in the last 2 years, mostly the first half year was logistics, and then it was a whirlwind of fieldwork and consultation with groups. So no analysis yet, though we're working on a few, I think, between students. And so we're working on a couple of papers, but nothing on the large scale. And yeah, always happy to have a chat on the approach on how to do it. I've got some ideas, but we'll see where you go from what actually happens with it.
Great. Yeah, and if anyone else has any other questions, feel free to pop them in the chat or Q&A, or raise your hand. I'll just wait a few more minutes. I've got a question for you, Maarten. I saw when you put up your violin plots, and it was like a marine score or something. Yeah. Lake was quite high.
Yeah, because what we're talking about is estuaries and not lakes. So there's not, and this is why it gets confusing, and I didn't want to get into detail. So these are terms, they're lakes that connect to the ocean, and they're generally large bodies of water just next to the ocean. They usually have at least one, sometimes multiple connections to the ocean. They're quite shallow, so there's lots of evaporation, and they're tidal driven. So they're marine, well, they're estuarine lakes. So they get quite high salinity. Some of the ones will be not quite marine, not quite 35, 36, but it'll be easily 30 parts per thousand, some of them.
So quite salty lakes.
Quite salty lakes, yes, definitely quite salty lakes.
Okay.
If there's no questions, I definitely want to plug, like, people have a look at the eDNA conference website. So the website is live now. So if you're interested to still help out, or just to get keen and get excited about it, have a look. Oh, there is, Samantha has a question.
Yeah.
Oh, that's a good question. I didn't think about that. I might have to acknowledge you on that one, Samantha. I'm gonna have to write that down. No, I'm not planning on doing that. It's a good idea. This is a good idea. We've got a few ideas on looking at, because in the end, what we want to do is basically people want to know, are their species getting better or worse? Like at some point, what's like the unique thing with this program, which I used to be purely marine scientist, because we often stand at the side of the road playing with syringes. So people obviously come up to us with questions, what the hell are you doing here? But people are very engaged with the estuaries because they're places where they live, right? In Australia, I think for the coast, pretty much 90% of people on the coast live within 5 kilometers from an estuary. There's over 180 estuaries in New South Wales. So people are very connected to it. They're places where they play, they go fishing, they go walking the dog. And so we get a lot of interest. And it doesn't matter if it's a recreational fisherman, whether it's an Aboriginal elder, whether it's the director of fisheries, everyone wants to basically know, is it getting better or worse? And there's so many different ways of looking at it. And one of the things to look at it is create an index of invasiveness or an index of native species, or look at how that ratio changes. This approach is cool though, Samantha. I'm gonna have a think about that. I'll definitely make sure to acknowledge you there though, because that could be fun to look at.
Then Anna's got a quick question about if you can comment on how you interpret the detections of unknown or unexpected taxa.
Oh, there's a fun one and a boring one. The unknown ones, there's basically not much we can do. So because we've got 10,000 taxa, the number of unknowns at the moment, it's actually pretty good. Depends what we're looking at. So with fish, because we got in Australia, the National Biodiversity Library, fish are at the moment, I think we're at about 70 to 80% is at species level. So we got pretty good resolution for fish. But once you go to bacteria, it's far less. So the unknowns, at the moment, I'm not doing much with it. I just basically see at what taxonomic level I can get them. And then it depends on the focus of the paper. There's a student working purely on fish. So then we can kind of look at just species level. And then Simone, who's looking at the flood impacts, we'll be looking at ciliates and bacteria. So then you start looking at different levels, obviously. For the unexpected species, and this is probably one of the fun stories, for the first set of data we got for the Richmond River, which is in the north of the state, we got water buffalo somewhere in the estuary. And I thought this is a mistake. There's some sort of contamination somewhere, but I couldn't rack my brain. How would you get water buffalo contamination? And after a long search with first myself and then my colleague Kate, we found out that 500 meters from the site, there was a farm that trained draft horses, and they used water buffaloes to train the draft horses. Basically, I didn't know this was a thing, but so just next to the site, there was water buffaloes. And apparently, for just a bit of too much information, they have very sticky poo compared to compared to cows, so their DNA keeps lingering quite long. One thing I'm very interested in, the very last site we surveyed was 1.5 kilometers downstream from a zoo, which has a gibbon enclosure just next to the water. So I'll let you know if we get gibbons and giraffes in estuaries in the south of the state, Anna. But so yeah, the unknowns, when I communicate it, and I think that's probably the real question, to communicate it to people who are not used to eDNA, one thing that helps me is to say what we're detecting is the DNA, the trace of DNA from the species, not the species itself. And a good example is when we sample from boat ramps, even in freshwater, if people drive their boat and go fishing, sometimes we'll pick up mahi-mahi or tuna, pelagic fish, and obviously they're not there. But what we see more often than not is that fishermen will discard their catch. So you'll take samples and you'll see just next to it, there's a frame of a tuna lying there. And you go, well, there's definitely tuna DNA here, but it doesn't live here. So and there's the interpretation. It's one of the questions that I've got in the end, and that's a challenge because there's so much data, is how do you interpret it? And that making that jump that it's the DNA of the species and not necessarily the species. So you still have to use your brain. You just can't go, yep, there's definitely nothing but tuna and water buffaloes in our estuaries. That can sometimes be challenging.
Indeed. I'm reminded of a colleague who found, I think, tomato in Antarctica, and it was probably part of somebody's sandwich rather than there being tomatoes growing in Antarctica.
I've had cabbage on coral reefs.
Yep. I think that's all the time we have for today. If you have more questions, I'm sure you can Google these people. Otherwise, find them on the SeDNAs website and feel free to shoot them an email. They're both very lovely. Yeah, and thank you so much. No, don't.
No, not true. Not very lovely.
Thank you so much for coming to this webinar. And we hope to see you again in June in our next webinar.
Bye-bye.
Bye.
Thank you, Claire.
Yeah, thanks.